So yeah, now that we're in a smaller group, I think this would be a good time to go around and give some introductions. So maybe Jason, we can start with you.
Yeah, hi, Jason Hilton. I'm with the Lattice project. We're relatively new. We're at Stanford University. We'll be doing the Data Coordination Center for the CZI networks. And just a little bit relevant to these topics, and at least initially, we won't be implementing our own standard pipelines or anything like that. So the data submissions will be raw data and processed data. So I'm really interested in understanding what the minimum is that we think we can ask in terms of QC metrics, or what we should be asking for as part of submissions. And then also, if we get into it, in a QA that we could do to verify provenance, that processed data X came from raw data Y, and so on.
Mm-hmm. That makes sense. And just for my own understanding, I think I missed the session giving the background on the Lattice Consortium. Could you just say a word or two about what that's doing?
There was no session. My introduction was the
session. Oh.
Yeah.
Okay. All right. Well, yeah, I didn't know if it was in one of the previous seminars I was introducing.
We came up with the name last week, and
okay.
So here we are. Yeah. So it's the CCI seed networks DCC essentially. So it's kind of a spinoff from the HCA DCP, but we're going to be. We're still trying to figure out the whole data workflow. We think that we will wrangle the data from the seed networks and then deposit the raw data at a repository and an archive. Maybe that is the DCP. We're hoping to have an easy data flow between the two. And then we're also going to be working with CZI. They're developing their cell by gene tool into something that's more web-based and more of a data portal or exploration tool.
Okay, gotcha. Thank you.
Yeah, and we'll be kind of introducing ourselves more formally soon, hopefully.
Okay, that's awesome. Thank you. Richard, do you want to introduce yourself?
My name is Richard Conroy. I work at NIH, and I'm the program leader for the HubMap program and a bunch of other programs. But at least for this effort, it's HubMap. And I think I have most of the people on the call before.
Yeah, we're probably starting to jump into similar sessions across the thing. I see C. McCollum. I don't know. Sorry, I'm not sure what your first name
Chuck.
is.
Sorry. Yeah, I'm web developer with HubMap. Looking a little bit at the ingest and how much QA we should, like how much automated stuff we should do, or should we just let data come in and then worry about it after the fact? And
okay, gotcha. Thanks. Alex?
Yeah, I know most of you on here, but Alex Roplewski, I'm the Director of Biomedical Applications at the Pittsburgh Supercomputing Center. I'm the PI on a data archive funded through the BRAIN Initiative focused on microscopy data. And I'm also participate in the HubMap project. And one other project that many people may not be aware of is I'm also participating in privately funded genomics DCC for the Aurora Breast Cancer Research Project.
I see William Sullivan.
Hi, I'm William Sullivan. I'm from UCSC, and I'm kind of associated with the HCA and DCP.
Okay, cool. And Takeshi?
Yeah, I'm Takeshi Saito working as a pharmacy. We are exploring to use the single cell RNA sequencing for the clinical application.
Okay, awesome. Thanks for joining. All right, great. This is a nice sized group. Hopefully we'll be able to figure out a lot of things here. So yeah, I guess we have more discussion prompts now that we are potentially tackling more topics, but I think there are some good questions from each box that maybe we can figure out how to tackle first. Maybe we can start with just talking about some of the common metrics, and then we can that can lead into how, whether or not those metrics are bespoke and, specific for consortiums, or how much they can be standardized across consortiums. So maybe we can kind of tackle both of those at the same time in a way. And I think this is also interesting because this question is interesting because it might be different for different data modalities. So whether or not you're doing single cell RNA sequencing or some sort of imaging based thing, or maybe even spatial transcriptomics, it's going to be different per assay. So I don't know if we have expertises from the different assays here. Maybe we can sort of go through each assay and talk about some of the metrics and how applicable we think they are across different consortiums. Does anybody want to volunteer a particular assay first? Or
so I'm going to volunteer Alex here. So I think we can talk a little bit about maybe imaging, QC, and metrics, and then also I think sequencing ones, because I think those are the two areas that seem to be the best developed, and at least in terms of traditional microscopy and RNA sequencing. So those might be good places to start. And the way we talked a little bit about this yesterday was in terms of general QC metrics that are sort of more information based, things like signal to noise ratio versus things that are more data type specific. So, what's the dynamic range in an image? So I don't know if there are, you guys probably know much more about this, but are there ways of trying to categorize some of these metrics a little bit?
Well, I guess my thought on this, and this also kind of goes back down to what we mean by QA and QC. In my mind, really, when we're talking about QA, we're really talking about is the data that you've received, without looking at the specific type of data that it is do you have everything that you need? Has the data been transferred properly? Are there no strange characters in the file name that'll cause pipelines problems down the road? Do checksums match up? That type of information. Can you read the file? If you know it's a TIFF file, can you at least open that with and get the metadata out of the TIFF file, for example? So those types of things is really what it's sort of the first step, the quality assurance step. The quality control really is in my mind, it's more the more modality specific. For example, in sequencing, there's a number of different common tools that will go in and assess the quality of the base calls for every read and give you some summary statistics based on that. For some other modalities, there are different ways that you can also provide different kinds of summary statistics as well. But all the ones that I've seen are really modality specific. So I think that's kind of where the dividing line is.
Your point about QA is actually really interesting, and I don't think people think about it a lot. They always tend to think more about just what the QC metrics that they're going to generating when they're analyzing their own data. I guess maybe the data coordinating centers are probably thinking about it more than individual sites. You mentioned things like checksums for files, making sure that there's complete metadata. I'm curious, maybe from the HubMap perspective, how are you guys going about that? I know a little bit from the HTAN perspective how they're sort of thinking about it, but yeah, I'd be curious to see if that's.
Well, I can just briefly update you on where we are with the HubMap project. The HubMap project has a lot of different data modalities. And what we've been doing recently is we've been having some sprints that are focused on specific data modalities. As a part of that sprint, we have experts that are generating the data, as well as folks at the DCC, or what we call the infrastructure and tools components. We're kind of organized a little bit differently than most genomics projects, for example. But what we've done is we specifically, as a part of that exercise, went through and defined the metadata that was specific to that individual modality, describe how the our central organization called the Hive is going to process that data and make it available. So how that specific processing pipeline works, and then then finally, which is actually before things get to that, we also talk about the QA/QC metrics that are appropriate for that particular data, and those are mostly the QC metrics that we're talking about.
Yeah.
And we have some draft documents for a couple different modalities. One is attack-seek, and the other is codex. The attack-seek document it will be similar to what you would do for any type of genomic sequencing, although there is some specific attack-seek information in there. And the codex document that we have, it's specific to codex, but there's also things in that document where we talk about QC that are applicable to a lot of different other imaging modalities as well.
Gotcha. And just out of curiosity, is there going to be like centralized pipelines that the HubMap is going to process all like data of a particular modality through, so it's all standardized, or are you letting individual centers or sites sort of do their own algorithms, their own pre-processing? It's just when they upload things, they have to have the complete metadata and the files in the correct format.
At least for the first data release, it'll be a combination of both. If there's a particular data modality that multiple sites are generating data for, that's where we're going to focus on having that pipeline and the standard processing of all that data together, so that when we do the release, all that data has been harmonized in some way according to the pipeline. There's also a lot of these different data modalities that only one site is producing, and at least for the first data release, we're going to make available what the data generating site has how they've processed the data. But we hope to have all that information as well documented.
Yeah. I think it sounds like we took a pretty similar approach in the HTAN, where it was going to be hard in the beginning to force like a particular way of doing things on all the individual centers, because each one already had their own sort of favorite way of doing things. And so I think the way we largely did is we allow them, I'm thinking mostly about the sequencing data here, but they generate it, they sort of align it and pre-process it however they want to. It's just, we have, we expect them to upload FASTQs with particular metadata, or, BAM with the alignment stuff. The one place where we did sort of align on a central pipeline was the generation of QC metrics. Is that like once you get to sort of like a raw counts matrix for like single cell RNA-seq, then there's a systematic set of tools that you can apply to that to generate like a complete list of QC metrics. And so that'll be uniformly generated. But then after that, basically all the different kinds of analyses that you can do on top of that, normalization and the clustering and all that stuff, is also going to be very specific. So then we just, left it at, allowing individual institutions to do it as long as they upload the data with the appropriate metadata.
Well, one of the questions that I have for you, and this is based on my experience with the Aurora project, which the Aurora project is really decentralized to the extent that there's sort of no overarching organization. I mean, the group has worked together for years, but for the DCC part, which the PSC is doing with the University of Pittsburgh, we originally had wanted to capture all the pipelines that people were using and containerize those pipelines. That has been an exercise in futility, mainly because getting cooperation from the data contributors to give us all the details that we needed to be able to repeat the analysis, it's been a real struggle. I mean, my recommendation really, if somebody's starting out anew, is to centralize as many pipelines as you can, because that way you'll what the provenance is when you start from raw data to higher level data. You know exactly what's been run, exactly what versions of things have been run, exactly what reference genomes have been used. You have all that information. And if you're collecting that on the back end and relying on other, the data contributors to provide that, some are very good at providing it, but others are not so good at providing it. So that is a danger with that approach.
Yeah. And as we're thinking about going cross consortiums here, right, each consortium is, even if each consortium has its own sort of gold standard pipeline that they want to apply all the data, now we got to try to go across consortium somehow. And so I think it'll be even harder to implement a single pipeline for each data type across consortiums. Wondering if a challenge we could try to think about in the next session, try to answer is how to go about finding consensus of sort of the minimum set of data, either quality assurance metrics or QC metrics per data modality that we would like to see be collected across different consortiums to at least help establish that.
Yeah, I think that's reasonable. The real issue with a lot of this stuff is, ultimately it's going to rely on somebody who has a code who implemented a method, and just like with some of the sequencing methods, where with QA/QC, where what they're really doing is they're not doing the QA/QC on the entire data set, but a subset of the data set and showing you that result. I think we need to be aware of these issues that the equivalent metric from different programs may be slightly different. So
yeah. Right, that's a good point. There's how people name things, and then there's what they actually calculated, and the two may not always be sort of concordant. They may name things the same thing, but calculate them differently. They may calculate the same thing, but name them differently. And so that can be challenging.
So I have a question. Alex, you mentioned the genome references, and that's been on my mind a lot, because even if you have slightly different pipelines, maybe the raw matrix files could still be combined in such a way that you could do some integrative analysis. You could get into how similar do the pipelines need to be and all that. But if you have the same transcriptome reference, then you still have the same columns in the matrix. So when you're accepting processed data, say raw matrix files from a variety of different labs and their own pipelines, are you getting agreement on what annotation version to use? Or are you just trying to track what they're using, or trying to bridge?
So in this case, we're lucky in the Aurora project in that they're all using the same genome reference. But with some earlier projects that's been an issue. Even with the current reference genome, sometimes certain groups go in and twiddle with it a little bit because they think they know a little bit more about one particular area, and while they say that that's the genome that they're using, it's not really the genome that they're using. But a lot of these issues are really hard to catch without, you know, going way down the pipeline and then finding out that some analysis is off. So yeah, there's a lot of, you know, there's a lot of these issues that are out there that I don't think there's necessarily a good way to handle.
Yeah, if I can channel my DCC members, it's like that's like the one thing that they really wanted to fix up front, because then it just makes their life so much easier on the back end. Is just choose a common GTF file for gene annotation and a common genome, and then it makes a lot of things simpler throughout the whole entire process. Otherwise, you have to deal with mappings and what versions of IDs they're using, and it's just every bioinformatician hates doing that stuff, even though it's probably 50% of what they end up doing. So, I mean, that would be something pretty substantial if we could figure out a way to use the same genome reference and GTF gene annotation file across consortiums. That'll probably make, you know, save thousands of work hours, you know, for individual groups trying to combine data across different things, at least for the sequencing. I don't know. Yeah, is there an equivalent when it comes to imaging, or is that just a whole different beast?
Well, it's a whole different beast. It depends on where people are going with imaging. So if you're, you know, let's say if you have a whole brain volume, one of the things that typically is done to get from 2D slices to a three-dimensional object is a lot of the techniques rely on a reference atlas that they can align sections to and morph the sections so that they can better assemble that volume. And there are multiple atlases that are out there. The Allen Atlas is one that, well, the Allen Atlases, I should say, are used frequently, but there are other atlases out there. And there are also other, you know, there's a lot of things out there that the atlas approach doesn't work because we don't have atlases yet. You know, it's one of the problems we're facing with the HubMap project is that we need essentially a small atlas for each tissue that we're dealing with, and some have really, you know, some are farther along than others in that regard. But that said, there's, you know, if you're just talking about sort of a two-dimensional image, you know, it's a little bit, you know, different problem. Yeah.
What if people? We got 5 minutes here before the break, and then when we come back, you know, we'll maybe pick a few problems to try to dive into. I think there's one really interesting prompt in the Bespoke QA sort of standardization versus standardization, where it's, you know, given different biology, you know, may require different sort of QA, QC metric protocols. How do we write QA, QC standards that allow for sort of flexibility, you know, I guess it says for the needs of biology, but do not inhibit a level of standard processing and data set integration. So I guess, yeah, it's more of a meta question of like, how do you even go about writing, you know, let's say we want to take a QC metric and we want, for single cell RNA-seq, we want everybody to calculate total number of UMIs per cell. What process do we go through, or, you know, how do we come up with standards and, you know, conventional naming and conventional way to calculate it to be able to do that sort of across consortiums? I mean, that's like the easiest probable metric that you can think of, but, you know, as the metrics get more and more complicated, I'm sure it's, you know, it gets even more and more challenging.
Yeah, I agree that's challenging. I mean, one way that occurs to me, this is partially related, but not totally related to what you were just saying. One of the approaches that HubMap is taking, and maybe Chuck can speak to this in a little bit more detail, but, you know, at least as far as metadata is concerned, what they've done is they've defined different metadata levels, where some are more general and applicable to large sets of data, and others that are more specific to to data. So you can imagine using sequencing as an example, or let's say any modality, you'll have sort of common, some common metadata for, let's say, donor tissue, for example. And that would be common to virtually all the different modalities. You'd collect, that type of metadata. Then, if it was sequencing data, the whole wide range of sequencing, types of sequencing that you can do, there's some metadata that would be associated with those. And then to the specific sequencing modality itself, there'd be even more data. So it's kind of this hierarchical categorization that may be useful in this regard.
Yeah, that's a good point. Right, we have something kind of similar where it's like there's like the required stuff, I guess, and then there's some optional stuff where it's like if people have additional sort of things that they want to upload, they can. I guess it's maybe a little bit different than what you're saying, but yeah. Okay, so we got about 2 minutes here, and then we'll take a break. Are there any other major issues or challenges that people think need to be addressed? I guess or any other discussion prompts that you think are really interesting that you would like to discuss more?
Well, I think one of the things too, and to get into our next session a little bit, or if we're thinking about this, it's like a lot of these problems that we've been discussing during this workshop seminar, is that there's one of the problems that we're facing is there's no inventory of what other people are doing that we can rely on. So it's almost as if the first step on any of these projects is we have to go back, we have to query everyone and really get a good inventory so that we know where folks are and what's in common, what isn't in common, and what the best practices seem to be amongst the different consortia.
Yeah, I totally agree. It seems it's probably going to be a common theme coming out. Yeah, I can imagine the same thing when we talked about the file formats yesterday. You could probably apply an similar process to the standardization of the QC metrics across the consortiums. First of all, thank you, scribes, for taking some awesome notes. You guys are doing a really good job. I was just reading through them again. And so now I'm down in the breakout session part 2. And I was just trying to copy some of the how might we's from the above, just so less scrolling for me. And so just summarizing some of the points that were made. So right now I have how might we decide on common QA metrics for each type of file and the associated metadata with that. Number 2, just how might we decide on common metrics to calculate for each data type at different stages of processing? How might we describe standards for calculation of each QC metrics? I mentioned the total UMIs before. Something like that could depend on, if they're just using, again, this example comes from single-cell RNA sequencing, where it depends on the underlying count matrix that they're using. The actual calculation is simple. It's just a column sum of the matrix, but they filtered out some rows of that matrix, or, depending on the GTF file that they use, it could actually come out with different calculations of total UMIs. And so even though the calculation is simple, what goes into it is important. So there probably should be some standardization there. How do we finalize common naming conventions for each QA/QC metric? How should we decide on common references to make the QC calculations consistent? I'm still going through some of the points up above. Does anybody else have any other key points that they would like to bring down from the first session, from the first box, that we can discuss?
I don't see where you're copying those to. Which section are you copying those to? Maybe
sorry,
internet's just slow.
Yeah, no worries. It's so there's the breakout session part 2. There's the big title, and then I started just a simple list pretty much right underneath that. So the starting point of part 2 are the how might we questions that this group may wish to take forward. The goal of part 2 is ideas and recommendations for cross-consortium collaboration. So right underneath that, I just started a list. I was just trying to kind of simplify the list from above into some key points that we would address.
So it's in the white, not in the green. Okay.
Yeah, sorry, it's in the white. Yeah, not the green. I think, yeah, the green box is for, we can start taking notes for recommendations. So I didn't want to put it there. And then the blue box is sort of our final template for what we want to do. So.
Can I just jump in and say something about that blue box?
Yes, please.
I know you're not there yet, but when you get there, just to really focus on, and I'll put this in the chat as well, you know, really to concrete actionable ideas, you know, that really are exciting and have kind of a wow factor, and, you know, something where you really think that you'd want to move it forward.
Yeah, I think it's hard to have a wow factors when we're talking about QC metrics, but I definitely agree that some solid action items are appropriate.
I put that in your
the thing that yeah, okay, thanks. Well, we'll do the best we can to snazz it up a little bit.
I will. I'll just put a few notes in. Go ahead.
I liked what we did in one of the other groups where it was like we sort of came up with a general roadmap, sort of like here's the big picture of all the steps that we would have to take to get to the end goal. And then we had some sort of immediate action items, immediate next steps that we can take to get the ball rolling. So hopefully we can come up with a set of those here.
Great. Terrific. I'll give you some recommendations here and then I'll get out of your way. Let me know if you need me.
Awesome. Thank you. All right. Do you guys want a minute to look through the list above? Is there anything else you want to include in this little summarized list? Mini summarized list.
Josh, I'm sure it's rolled into one of these, but you're thinking through, okay, so we're generating all of these metrics. How are we going to share them? In that, you know, are these going to be posted on the data portals alongside the data sets themselves? You know, how are we going to share these metrics in a way that people can then use or interpret, or, you know, kind of what's the purpose for doing this?
Yeah, that's a good question. And that maybe overlaps a little bit with what the other group was doing. They were talking a little bit about sharing, or that it'll be interesting to see what they come up with in terms of mechanisms for sharing. I think, yeah, there's like the, mechanisms to make those sort of pipelines themselves shareable and get them adopted sort of by the different communities. But that's, you know, I think that's certainly something we can discuss too and think about as well, and then go see how we sort of jive with the other group. Right, it's an interesting point. I mean, my lab's been thinking about this, and as the member of these, working on the HTN, we decided on exactly this one key point where it's like, okay, we get the raw counts file from every group. We know what that looks like, and we know that we can calculate a bunch of standardized QC metrics on top of that. So we ended up writing a pipeline that calculates, I don't know, about 20 or so, 25 different QC metrics, using a combination of tools that are already out there. And so then we know that we are going to have a consistent set of QC metrics on every data set, even if the upstream pipeline for generating that counts matrix is different for each individual institution. So that's one way, at least at one key sort of bottleneck in the analysis, or one key hub in the analysis, that we could do something automatically consistent by just running the same thing, or having everybody run the same thing on that counts matrix. So that might be one way, is, you know, where standardized pipelines would be really useful, is like if you know the data is going to be in a key format, and you know that there's not a lot of subjectivity in calculating those metrics. Like some metrics are really hard. They require some, user observation and tweaking of parameters till they feel like they got like cell clustering in a way is like that, I guess it's not QC metric, but there's not like a universal approach somebody can just automatically imply and get the right answer each time. But for metrics that are more standardizable and set in stone, I guess, I think that's where it becomes really useful.
So I don't know if this is the right way to think about it, but it seems like some of these metrics are kind of internal quality checks of what's coming in at the point of ingestion. And, maybe to what Alex said earlier, those are the quality assurance metrics. And then there's quality control metrics for different parts of the pipeline. And maybe, again, some of those are for internal checks about the way in which the analysis pipeline, for example, has been applied, whether, something about the potential reproducibility of any data that's been generated given the metadata that's available about it. So, it may meet the minimum requirements, but there may be fields missing that may make it more difficult to reproduce. Or, it could be something about, for example, the use of antibodies and whether we think the antibodies are reproducible. It seems like there's some of these are more quality assurance and some are more quality control, and depending on what it is, we may want to think about it in a slightly different way.
Yeah, I totally agree. I think the quality assurance things, making sure that the underlying files have the appropriate integrity and that they're the files that users think they are and that the data coordinating centers think they should be, is important. There's the metadata that goes into how different QC metrics were generated, what pipeline we're using, what version, what references, that kind of stuff. And then there's the actual QC metrics themselves, like how are those being calculated and shared and stored and whatnot. So I think those are all kind of different. It seems like a good point that all those are important.
And maybe, again, this is just an assumption I've made, but it sounds like you guys are planning and generating some kind of quality metrics for all data sets. And Alex, I just wanted to check if that's also the same in Brian, and that, quite often we've talked about different levels of data. And I'm just wondering if, essentially if there are different quality metrics associated maybe with different levels of data, or is that not the right way to be thinking about this?
Yeah, I think that is a way to think about that. I mean, we had a discussion earlier in a session that you weren't a part of, Richard, that even the data states between projects doesn't quite isn't quite as well defined as it should be, and if that's also another thing that needs to be resolved before you can actually say, okay, if this is at this level, then you need to do A, B, C. So it's really, I don't know. It's all kind of how we describe this, and we really need more information about what everyone is doing, and it's kind of a collaborative process to rope everyone into, kind of using the same nomenclature for similar things.
Yeah, no, I agree. That's the problem. It's interesting that you mentioned data levels too. Because that's how our group has gone about sort of doing things one step at a time, is to sort of define a data level. Here's level 1, the experimental metrics and FASTQ files. Here's everything that we want to collect about those. We work on that. And then like level 2 is, the bands. Level 3 is whatever, count matrices. But the DCC, they don't actually care about, in our group, they don't actually care about the levels. Like there's nothing in their system that keeps track of this data type is level 1, this data type is level 2. It was just a way to conceptualize for us, like what we were working on at that time. But I definitely agree with your point that it's the different data levels have different things associated with it. and some are, it's a lot easier to come up with sort of systematic metrics, and whereas other ones it's hard. At least with the single cell RNA-seq stuff, it's, you know, the pre-processing pipelines, there's just a wide variety out there, and what metrics they produce and don't produce is sort of fairly different. And so we found it harder to try to standardize that up front. I think what we're going to do is something similar to, I think maybe what Alex was saying, what you guys were going to do is, you know, we're going to have all users sort of upload the raw data as well as their pre-processed data that they use with whatever, you know, they wanted to as sort of a first release. And then once all the data is there in a centralized place, it'll be a lot easier to go back and, you know, a small group of people just to rerun everything through a central pipeline. And then you come up with like a second release that's like, okay, this has all been, you know, run through the pipeline, that one sort of. So I think that's the approach we're going to take. I guess that's another idea too, is a way to enforce or potentially have standardization across different consortiums too, is that I'm assuming all the consortiums will have their raw data get put up into their own repositories. Maybe at some point a universal pipeline can go through and rerun all, you know, data from all repositories together. And then that would also produce a uniform everything. But yeah, I don't know if that's feasible in the near term, just because it seems like every consortium is still just worried about, you know, getting their data in the first round. So that may be something more long term. Does that answer your question?
Yeah, and I think where we're headed with this, if we wanted to kind of think of an overarching goal, it's really what we want to do is to be able to define best practices for what do we call them, data coordinating centers or data distributors or data archives of various types to follow.
Right. Yeah, that would be nice, actually. Right, because it's going to be hard at this level at this time to really kind of force different consortiums to do the same thing. But if we can come up with recommendations, and then hopefully the different consortiums can try to adhere to them, then it'll make it easier in the long run. So I think that's a good point. And a good word to use, recommendations, instead of requirements or something like that.
So maybe to run with that, is there a particular recommendation we might want to try to flesh out around recommendations? Or, you know, what if you had to think about a way of, first of all, getting together what those best practices might be, and then, you know, how we share those across data coordinating centers? You know, how would you see that process coming together? And is there an obvious place to start? You know, for example, RNA-seq is done by nearly all of the groups, if not all of the groups. And, you know, I think to Josh's point, at least my impression is although there are diverse pipelines that people use, you know, most of the data formats exist within a small universe, and the analysis pipelines, you know, is a slightly wider universe, but still it's manageable to wrap your arms around. So is that something we might suggest as a place to start here?
Yeah, that's fine. Yeah, I'm curious for the imaging perspective. It's probably a lot harder in terms of, it's probably the wild west in terms of pipelines and whatnot. Are there, again, I just don't know very much about it. I'd be curious to see, are there like points where it's like, okay, you get pre-processed data and there's like a sort of a limited set of formats that pre-processed data can be, or is it like, you know, there's just hundreds of formats out there, or, you know, how crazy is it?
It's pretty much the Wild West. I mean, things are getting better, but every microscope tends to put data out in their own proprietary format, although there's a few microscopes that have widespread adoption in the community. There are some open standards. OME-TIFF is one. There's also some other file formats that are intended to supersede what OME-TIFF does, but those are farther out. No, so yeah, it's a lot more difficult too with imaging too, because we like to classify everything as imaging, but really imaging is a lot of different things. There's codex imaging, which while it's imaging, it also kind of gives you different information than if your goal was to create a three-dimensional volume. There's imaging as well in spatial transcriptomics, but it's more on the order of spots in a location. So yeah, it's imaging is still the Wild West. It would be great as a goal if we could address that, but I think the easier way to proceed is probably on the sequencing side rather than the imaging side.
Yeah,
I think
that makes sense. I wonder. Oh, go ahead. Sorry.
I was going to say, at least on the imaging side, I think there are a number of basic QA metrics, such as the amount of contrast that are present in an image. Is it saturated or not? There's a bunch of sort of simple characterizations of an image that are independent of the content of the image. That in theory, that we could at least apply those and generate some common set. But I mean, I agree with what Alex said. I think it's hard to come up with the equivalent in the imaging space that I think we can get done in the sequencing side, because of just the variety of different types of imaging out there. So I think there may be a case of picking what's the most common type of imaging if we want to make any headway at all, and maybe codex being the potential example.
Yeah, makes sense. And I wonder if we can use the sequencing to sort of define a procedure or like a roadmap for how to go about coming up with some consensus, and then that can be used to sort of plow the way. Imaging can maybe try to follow that similar procedure or something like that. I don't know how much parallel there's going to be. Maybe it'll be a totally different roadmap, but so that all makes sense.
I mean, maybe it makes sense if we, as a part of this exercise, to address a sequencing modality and an imaging modality, and maybe use codex as the example on the imaging side. That way there'd be a roadmap for that either side would be comfortable with, let's say, if we wanted to move this out and expand it.
That's a really good point. I like that idea. And do you think the same type of, do you think the sort of QA and QC measures can be handled at the same time? Or do you think they need separate? I I almost wonder if the QA, the quality assurance metrics, some of that almost goes more into like the file formats and the metadata discussions. I guess it sort of overlaps with us and with those types of discussions.
Yeah, I mean, maybe one of the things that we do is we just think of the bigger picture. I mean, we're going to have, it's like what the comment that I made earlier. We're going to have a lot of these discussions that the first step is always going to be, let's see what other people are doing and kind of collect that information in one place. And whether we're talking about file formats or metadata or QA QC, it's essentially the same process. We need to identify a few key projects and collect the measures and see if we can come to a consensus on the measures with the smaller group, and then bring in additional people over time and grow it out. I don't know. That's my thought.
So how big a group would this need to be? And
to me, it's the important thing is to have representation from each, a knowledgeable person from each consortium. So however many consortiums there are that we want to try to tap, that would be sort of the minimum number of people that I would think would make sense. I mean, it can be more, right? You can have more, multiple people from different consortiums if that helps. But there, yeah, I think that's the major goal, is to try to build consensus. I guess maybe, some consortiums are smaller, maybe they don't have as much resources to dedicate extra time to, worrying about this stuff. So I guess, maybe there are a couple larger consortiums that maybe could take the lead on it, and then be optional for some of the other consortiums to sort of come into. But
well, and I'm going to be frank about this point, because it's been emphasized to me by my program officer on the BRAIN project, is that, and as each, and this is this is no surprise to Richard, because he works at NIH, but NIH is really focused on their particular area. And while my program officer thinks it's great that we expand what we're doing metadata wide with a bunch of different consortiums, the real focus has to be on BRAIN. And that's where that box is fairly tightly drawn. So the amount of resource that you can devote to all these other things that would make your data 10 to 100 times more useful are not given as high of a priority by the funders as what they should be. But I'm gonna get off my soapbox here.
No, that's totally true. And I mean, yeah, I'm not officially technically a part of our DCC and the HTAN, but it's the same thing. They're swamped just trying to get it up and running just for the HTAN. So after this, when I go back and tell them, hey guys, guess what? You get to do everything that you've already been doing, but again, with more people, they're gonna shoot me, right? So, but I wonder if that's also a recommendation that we can make to the higher ups at the NIH, people like Richard. No, but it's where, if they really want this, because it will be useful in the long run, then seeing if there are ways to put some resources behind it, because I think that's the biggest challenge, is it just takes people's time and so maybe they can come up with, I don't know, what sort of grants or mechanisms or supplements or something like that could try to facilitate these things.
Yeah, I agree, and it kind of has to be, either multiple agencies or from the Common Fund or something. And I know that the Common Fund is trying to do that with a lot of the Common Fund projects, but there's other projects out there that would also be useful to collaborate with and
yeah. So maybe that needs to be a recommendation. We should just copy and paste that recommendation on every single, every one of our working groups that we've been a part of, because to make sure the message gets across.
So with that in mind, could you, given that, first of all of you have limited time. Secondly, all of the consortia are busy doing their own thing and trying to develop solutions in real time for what their challenges are. So, it's not just a question of providing money for people's time. It's more than that. It's also this challenge of different consortia or different stages of development and have different modalities present in terms of the data they're generating. There's lots of different situations for each consortia. So, again, are there more obvious places to start than others? And, it seems like if you think about it from the perspective of use cases for why we would want to do this, why we would want to have common quality metrics, are there particular use cases that would naturally drive together particular consortia to want to have a common QA/QC infrastructure?
Yeah, no, I think that's a really good point. If we can come up with specific examples, that would help make the thing a little bit more concrete. I like the discussion we had earlier about the reference, and like I'm just thinking, like people will want to do meta-analysis of their single-cell RNA sequencing data sets across consortiums, and just to be able to combine data sets if you're using the same underlying gene reference, so that way that it just makes everyone's life so much easier, and it will save a lot of time on the back end if you can do that on the front end. So I'm wondering, that could be a simple recommendation that comes out of here on the single cell RNA-seq side, is that the reference files people use, the genome build, the GTFs, that stuff like that, we can come to an agreement early on and try to enforce that in the different consortiums. It's just a decision that everybody has to make. So you just get everybody to make the same one, and then that will, have a lot of nice things, nice impact downstream. I don't know if there are other simple examples like that either, or like with the codex, if there's simple things that we can make there as well.
Really having a common QC metric would make it easier for the researcher to identify data that is relevant to whatever use case they're looking at, because everybody's interested in the best data that's out there. And sometimes that's hard to determine from, if you go to 10 different places to get the data, you can't, you necessarily have to spend a lot more time figuring this out if they're using different metrics than if similar metrics were available. One of the other issues that was brought up in one of our sprints, particularly with QA/QC, was that there was the folks generating the data were okay with kind of having QA/QC metrics associated with their data, but what some of them did not want to do is to kind of say, kind of give a ranking according to that, because different researchers may feel different metrics are more appropriate than other metrics. And they were comfortable with providing those metrics, but they didn't necessarily want the data presented in a way so that one of these metrics was the master metric.
Yeah, I dealt with a lot of this as part of the ENCODE DCC, and so you want to report QC metrics, right? So then the data consumer that's aware of what metrics mean what, they can come and they can figure out what they want to use based on what metrics. And you have the sensitive data generator that doesn't want, at ENCODE we use different levels of audits to say this is, this is in this kind of caution, use with caution area, or this is below the threshold. We really don't recommend it. And they certainly, data generators certainly aren't very easily agreeable to that type of thing. But then you have the data consumer that doesn't really understand or have opinions about the QC metrics, and they just want to know, this data that I'm looking at, is this good? Should I use this or not? And so that's where you want to get into some thresholds or have some generalization to say, yes, use, no, don't, whether it's a ranking or not. And it depends on also your data set. At ENCODE, we had maybe 5 or 6 RNA-seq experiments on K562. There you need the ranking. You need some default data set to say, if you're interested in this, use this one. So it's a delicate balance because you want thresholds, but then the data generators don't want those thresholds, or they have opinions about the thresholds as they should. So it takes a lot of buy-in.
Yeah, no, I totally agree. We in the HCA, we were going to do it, sort of. We're going to give a lot of power to the individual centers to kind of decide what, how they were going to do their filtering, and basically what was going to go into the publication. That's really what we wanted to make sure to capture on this first round. So even if we didn't necessarily agree with, exactly what threshold they were going to use, or as long as it gets published and is okay, we want to be able to regenerate the exact same figure that they had in their publication for users, because that's oftentimes what they want to do. They want to go, you know, look at the same UMAP that's in the paper and plot their own gene on it, or something like that. So that's why we haven't been very strong on it so far in terms of enforcing particular quality metrics. And it's also hard because we have so many different platforms. Some samples are just a lot harder to get and harder to process, so they might have lower quality, but they're so valuable because of the clinical specimens that they're coming from. And we deal with clinical specimens a lot, so we don't necessarily want to, you know, poo-poo on those data sets just because the samples are harder to get and process. So yeah, it's a challenging problem.
So I kind of want to go back to one of the points that Richard had raised earlier, and that is, you know, who do we start with? You know, I'm with HubMap, and I'm with Brain on the imaging side. We're focusing on Codex. That's more HubMap than Brain-wise. You know, I would be you know, given my limited amount of time, I'd be able to devote a limited amount of time to kind of help to take this forward. So that would be the HubMap Consortium. Would HTAN be willing to participate in? I mean, can we identify 2 or 3 groups that might be willing to get started at this point, or do we need to beg people at the end of the consortium, at the end of the workshop or whatever this is, and say, please, we need volunteers?
Yeah, yeah. I mean, from the HTAN, I'd be happy to participate. I can probably get 1 or 2 of the actual people in the DCC to jump in. You know, as long as it's in the beginning, at least. If it's just like a call and like planning stages, then I think that makes sense. Yeah, it's if you're going to get into really time-consuming stuff, then obviously that's where everybody would probably shy away from it. But I think an occasional call, and especially like what you're saying, doing the inventory, which is such the first critical step, I think that's something everybody can do with relatively minimal effort. So I think that would be possible. I'm sure it could be possible across the HCA as well.
And it sort of feels like UMI count is something that everybody's generating anyway. And you know, given that nearly all of the consortia are doing RNA sequencing, you know, I mean, that just feels like, of all the things we've discussed, that's probably the lowest hanging fruit here. I don't know.
Yeah, no, I totally agree. I think it's furthest along. There's already common sets of metrics people use. I think there's additional ones that could be added, but yeah, I agree that it makes sense, right? I guess the one question is, if we're thinking about, you know, starting off additional working groups, do you try to keep everybody together across the different modalities? Probably, just to make it easier, but I don't know how exactly specific the imaging stuff will be from the sequencing, or if we just go with sequencing first and then, you know, come back to imaging later. But
well, I was thinking a little bit more about the imaging side, and Alex, please weigh in on this, because you know, you mentioned at the start the Codex sprint that happened recently. You know, a challenge with Codex, for example, is the quality of the Codex data depends on the quality of the antibodies that are used, and you know, there's a whole set of metrics that I think end up getting wrapped up into how we think about the quality of the Codex data itself, beyond just the details of what gets uploaded to each of the data coordinating centers. So that one, at least in my mind, seems a much more complicated problem to tackle, in that the quality metrics you probably want to track are a more complicated set.
Yeah, I think, you know, the whole issue of the antibodies that were used, I think what we just decided was that we were just going to, you know, maintain a list at this point and not really get into that whole issue. We were really concerned more with the images themselves and whether the images themselves were quality. I mean, that said, there's also another possibility that it's not as far along, and it's at some levels it's still more the wild, wild west. So this may not be such a good suggestion. Is the whole concept of spatial transcriptomics, and can that be brought in?
Yeah, that'll be really interesting. I think a lot, I'm based on the very limited experience I've had with it so far, a lot of the same metrics that get used in single cell RNA-seq can probably be carried over, but there's some additional things that would also need to be done as well.
Yeah, it really depends on how people are doing their experiments with RNA-seq, because ultimately if people do are trying to do these sort of multimodal experiments, well, you never quite get the same thing because you're not actually using the exact same cell, for example. You're at least in that same area.
Mm-hmm, right.
But I still kind of go back to and agree that the RNA-seq and some of the different types of RNA sequencing that are being done nowadays are that's probably the easiest low-hanging fruit.
Yeah, that's fine. And we can start there thinking about it. Again, thank you to the scribes here. I'm just looking at your notes. I keep checking back in occasionally, and you guys are doing an awesome job capturing suggestions and stuff. So yeah, I guess as I'm thinking about the RNA-seq, right, there's lists of things that can be done sort of now that are concrete action items, use same gene reference. And then there's other things which will take, that can be done now, that can just be suggested to all consortiums, to all groups. But then there are other things where it's going to take consensus building. And so then I think that's where the meetings come in, and doing the general steps are do the inventory across the different groups, see what's being done in common, consistently being done in common, seeing what's easy to sort of generate on standardized file formats. Like another example is like the FASTQ files, and running something like FastQC, which just calculates a bunch of metrics on just your sequencing data. That's like super simple, and everybody does it because everybody's already using the same file format. So there are particular points where we could suggest using common tools, common pipelines to generate a common set of QC metrics. And then there are other points where I think it's going to take a little bit more work coming up with the consensus.
On the genome reference or the transcriptome reference, do you think there's going to need to be the stipulation that we understand there's scientific value in custom annotation reference or something like that, but pick one custom and then also do this agreed upon reference, and then it becomes more of a UX situation for each portal to clearly display? Because it's getting that agreement's going to be really hard.
Yeah, it is. Although we found that a lot of the same centers were already using similar GTFs, maybe a slightly different version. And so I don't, it's like Ensemble ID's GTF version, whatever, I forget the version number is now, but we just had to decide on, like, was it the simple GTF or was it like the full one? But I think because that's very relatively little work for people, so that probably for people doing most of the projects who don't really care about the custom stuff, it'll probably work well, I imagine. But you're right, the custom stuff, I don't think, you can necessarily be sort of forceful in saying, well, each consortium is only going to take this GTF. We're not going to do anything else. And, if you happen to need something custom, sorry, something like that, that's obviously not an option. So it's like the more consistent you can make it up front, the better, but enforcing that as a sort of a hard rule, I think, will be difficult to those few researchers who do really need that custom thing. Or, especially now with all the viral work, people are wanting to add in like viral genomes. And so exactly, they want to count other things going on at the same time.
Yeah.
At the very least, it would be easy if for the human transcripts, human or mouse transcripts, they can all use like the same ID scheme, like Ensemble or something like that. So that way, at least subsets of the matrix will always be sort of easily mappable between studies. But yeah, you're right. You may have additional custom features which you quantified on top of that.
It almost gets to the different use cases, like you mentioned, recreating that figure in the paper, right? That may require that custom reference, but then when you want the use case of integrating across all these different projects, then you want the standard GTF.
Exactly. Yep. Are there any other sort of hard, quick recommendations we can make? And the GTF is the only one that really comes into my mind. I think everything else will probably have to meet and do some GTF and reference for mouse and human reference build. My gut is that everything else will probably take some sort of meeting and inventorying and consensus on, but if anybody else has any ideas for the sort of the hard recommendations. Okay.
Yeah, I don't have anything else to add.
Yeah. All right, that's great. So let's see. Yeah, so we still got half an hour left. All right, so maybe we can work on drafting our blue box here for the last little bit. These are all good recommendations in the green box. Somebody want to take a stab at it, or I guess I can just start editing it here if you don't mind a little bit of silence while I
For a
think about
title, I would suggest something along the lines of maybe best practices and recommendations for QA, QC.
It's a good one.
We can solve everything in the world. That's what we can do.
Yeah, right.
Seriously.
I'm just going to put, so I'm a part of HTN, I'm just going to say cancer. We can solve that.
Yeah, why not?
But that's it's solved so many times.
Determine.
So I think what one of the things that we can, if we wanted to think about this, what we want to solve, it's really enable researchers studying, everything under the sun. We put cancer in parentheses, but it could be, any real disease. Enable researcher, or even people that are even doing cross-species comparison or something. But we want to enable researchers to have a uniform view of data quality across consortium.
Right, that's a good way to say it. I'll just add in for each data modality.
Yeah, that would be fine.
I guess we can say things that this will be useful for: facilitating integration
Data
studies.
reuse, integration
Yeah.
studies.
Any other sub points? This one, what would that having a uniform view of quality allow us to do? Let's see. Yeah, that's probably the main one.
maybe one of the things too would be to identify, and this may be pie in the sky, identify benchmarking data sets that can be used either for algorithm development or even just other purposes. Maybe it is sort of a benchmark looking at QA/QC in a different light. I don't know.
Yep, that's a good point. All right, that's good. So we're moving to recommendations for, we'll subdivide it by single cell RNA-seq. This is probably true for ATAC-seq as well.
Yes.
I guess I can put here basic some of the basic QC metrics should be straightforward, like total UMIs. Pretty much everybody does that. And yeah, I forget, for like codex or anything, was there any immediate recommendations that'd be good to come to consensus on, or you want to save that for more of the?
I don't think there were any immediate
Okay.
recommendations.
Alex, I mean, I think that's something good for us to think internally about within HubMap. And I mean, I agree with you, there's nothing immediate, but it's certainly something we should give some thought to within HubMap, if not, no other way forward.
I agree, and it kind of goes back down to your point about the the antibodies and whatnot. So yeah.
How do even people just report antibodies, or keep track of versions of antibodies that they're using? Is there like a standard way of doing that?
There isn't, but we may have one in HubMap. I know whenever we had the sprint, another gentleman, Bill Shirey, and I talked with some of the folks who were producing the data, and we kind of come up with a conceptual model for how we can maintain a list of antibodies, and if something was replaced or superseded by something else, how we can tie that all together in a relatively straightforward manner. Unfortunately, I haven't followed up with Bill since then, so I don't know what the priority was. I mean, we're really trying to focus on getting our data release out. So there's a lot of these great ideas that are kind of on the back burner right now that we need to kind of at least keep track of and re-engage when we have the time to do it. But it is something that I think is broad and could impact lots of different consortia. Now, how that pertains to QA QC, that's maybe I don't know. But it is one of these things that we need to be aware of.
ENCODE has a pretty extensive antibody schema, and they also went through. ENCODE 3 was really heavy on validating antibodies, and so there was a whole review panel, and so they have standards written out and everything like that. So that in itself is a resource that ENCODE offers. Is this?
Yeah, and I think we borrowed some of that for HubMap, or at least everything from the fields you'd used as part of ENCODE through to some of the thinking.
Yeah.
But as you brought up, that the sticking point is quite often attaching labels to data and how some people interpret those.
Maybe one of the things to do here for an immediate recommendation is, or maybe this isn't an immediate recommendation, but it's really to identify things beyond RNA-seq that are in common that we need to, let's focus on a few things, not not everything.
Yeah, so I was going to ask, if you had to pick one thing to focus on first, that would have the largest impact in terms of reproducibility and quality control and stuff like that. What do you think it would be for Codex?
So I think there are some things like, looking at the mean and standard deviation of an image as a way of trying to get at some signal to noise ratio. Likewise, there are metrics out there for looking at whether an image is in focus or not. And I don't know how well these work across all the different types of imaging, and Alex might have a better sense of this, but I think there's probably a couple of reasons.
Yeah, but there's some things too, like if you do your Codex experiment in the right way, you'll have blanks, and then you can kind of compare those blanks and get doing some analysis, you'll get two peaks rather than a single peak. There's a number of different things that one can do. Some of that is actually how you do your experiment itself and how you're collecting that, because we found out as some folks are doing it, they're just doing the first series blank, and others are kind of doing blanks periodically throughout the experiment, so you can get a better view of whenever something has changed. So it really just kind of depends on how people do their experiments. And I mean, I guess that's another thing that we're not necessarily addressing here, but it's also another thing that impacts QA/QC, is that there's certain ways that people would need to do their experiments, and there's certain pieces of metadata that they would need to provide to be able to do some more robust QA/QC down the line. So, these are the kind of things, as we go through each of these different modalities, we would need to think about. Is there additional information that we would need to capture from the data producer, or have them do their experiment in a certain ways to enable, a certain QA/QC process to work?
So it feels like, at least for the Codex, getting together some small group of the of people who are really doing the codex, because I think at least for HTAN and HubMap, there's at least an overlap in the groups involved. So it feels like the codex universe is somewhat small enough, and there's a wider range of things to discuss beyond just QA/QC. So as part of the sprint that we did internally within HubMap, we talked about the analysis pipeline and other details. So I don't know, Alex, if you think there's much value in trying to bring in the other groups dealing with codex into that discussion, or whether that's just going to.
Well, I don't know what other groups are producing codecs. It would probably be beneficial, particularly because it's a newer technology and there aren't a whole lot of people doing it. So there's always the opportunity, if it's a newer technology, not a whole lot of people are doing it. But if you set the standard, then everyone's going to follow that particular standard, which I think is a good outcome, rather than having a bunch of different competing ways of doing things. So at the sprint, we've of course had the folks from Florida, from Vanderbilt, and from Stanford there, and they were, but I don't know who and what outside groups are necessarily dealing with codex. Perhaps you can point us in the right direction. It wouldn't be a bad idea to identify those people and begin the discussion.
So maybe that's an action item, is suggesting that the groups working on multiplexed immunofluorescence, in particular codex, that there's some kind of working group. One of the topics that they should deal with is QA/QC of codex data.
Yeah, I mean, we have, there's already probably a sub-working group, and most consortiums are already doing that. So it's just a matter of getting the subgroups that are already meeting to somehow meet together.
Or the leads and trying to
Yeah, exactly. So that's a good point. Maybe we should put that more explicitly.
Or even, where you have the even identify key persons. I mean, it's really key persons or data groups, whoever's.
Let's see here.
Hi, it's Maggie here. I'm just going to chip in for a minute. There's some messaging in the Zoom chat for you, just to let you know where to go when you come back from lunch, which is in about 15 minutes or so. All
right, thank you, Maggie.
All righty, good.
Yeah, just in terms of sort of the immediate action items or next steps. So get leads of, say, codex. Do you want to be specific to codex or just say imaging? And make it more general to all imaging platforms. To get them, see if we can get them at least talking to each other or connected.
So I think we want to keep it relatively focused to multiplexed immunofluorescence, because imaging in general, that includes things like imaging mass spec, and it sort of becomes more complicated. Whereas I think multiplexed immunofluorescence, it sort of keeps it focused on antibody labeled microscopy.
Mm-hmm. Do we want to try to get people to actually meet or just in contact with each other?
I think in contact with each other. Let's try not to be too ambitious about that.
Yeah, right. To determine if there's a path.
I mean, I don't think anyone at this point outside individual groups has generated a substantial amount of codex data. So, you know, obviously the Stanford group has done a lot, but beyond that, I'm not sure.
I mean, I think also one of the things that should be here in one of these action items, I'm not sure if it necessarily is immediate or later, probably more later, is I think what we need to do is we need to prioritize, you know, where we think the effort is will produce the best bang for the buck at this point. So I'm not sure how that kind of fits in here. But it's because I mean we've talked about a lot of different modalities, and the inventory will shed some light on that.
Yep.
But after that, we really need to kind of figure out where the focus is. You know, the next focus should be to take this further.
Yeah, I think right after the inventory step, you almost have to form, I mean, I just tried jotting something down here. You almost have to, that's when you have to meet again with some of the key players or interested players to, we're probably really
maybe
real
an
work
action
begins
item would be to organize a meeting of the key players at some point.
Yeah.
I don't know. To maybe to set priorities or something, you know.
Do we want to say an action item about sort of recommending to the funding agencies, see if they can help facilitate bringing together these working groups?
Yeah, maybe the point is to you know, incentivize, you know, because I mean, you know, if so, there's two parts of this. The first is the just getting people together and kind of agreeing on the standard. The second thing is having everybody go back through the data that they have right now and implement that standard. And both of those require resources. So
yeah.
Should that be part of the taking the inventory, is to figure out what metrics there are and look at the existing data so you kind of get a feel for what the general quality is and maybe use that as a guide?
Yeah, I think that's definitely the starting point. Yeah, the challenge comes, right, let's say you do that and then you see, you get some consensus on some metrics, but then maybe there's some that, some consortiums use, but other ones don't. And you come to a consensus, okay, all consortiums should use it, or, you recommend it or whatever, and somebody's going to have to go back and update their documentation and metadata files and stuff like that. So, which, I mean, it's fine. We're sort of planning on doing that in the HTAN. We're going to have versions for all of our metadata stuff, assuming that things are going to change over time. So we'll sort of have, we'll be able to accommodate that. But Alex is right. It just takes time. And so it's really just,
everyone might be able to time that well with when they're implementing a new pipeline anyway, or something like that.
Yeah. All right, how does that look so far?
So who would you want to do each of these? that's the part that
Yeah. That's the thing, right? It's easy to make the list. It's hard to put your name next to actually doing something. I mean, I'm certainly interested. I mean, that's what my lab does, sort of build some QC pipelines for RNA-seq stuff, so single-cell RNA-seq stuff. So I'm happy to be a part of that conversation. It's more of just, I guess the challenge for me is like, okay, HubMap, who do I connect to there? Like, let's say I wanted to meet with somebody in a couple of weeks from now. Is that you, Alex? Is there another sort of single-cell RNA-seq pipelining expert?
So there's a couple things. The first is that we did this sprint, and we documented sort of proposed standards, how we're going to do our QA/QC. That's in draft format. I don't think there's a problem, and Richard can correct me if he thinks there is a problem. I don't think there's a problem sharing that document with you, and so you at least have that knowledge to build. And really, with anybody in the entire HubMap consortium. So the group that I'm a part of is the infrastructure and engagement component. So we deal with basically everyone in the consortium. I can hook you up with the right person that you need to talk to. And if I don't know the right person, I know the right people to ask to get who the right person is. So.
Yeah. No, same here. We have I guess we call them RFCs, requests for comments, where we have a bunch of these Google Docs set up where we got these lists of metadata files for each sort of different data level of each data type. So we could easily just think about sharing that. Again, I'll just double check with my DCC about it, but maybe that's the easiest way. Somehow we've already done the inventory. I guess it's just a matter of sharing that inventory with the different groups. So I can imagine, HTAN, HubMap, HCA, I think they probably have theirs. We've been trying to coordinate with the HCA already. Sure, maybe you guys have too. So maybe it's just a matter of getting those groups together. And Lattice, if you guys have any too, you're certainly welcome.
And I mean, and the other thing I wanted to, so we can share with what we have right now and what we think we're going to get. We are going to have some additional what we call tissue mapping centers added to the project. And my understanding is that those are going to be, some of those are going to be completely different data modalities. So I don't have information on those, but that will be coming down the future. So it would be, from my own bias perspective, it would be helpful to know what everybody else has so that we don't have to reinvent the wheel for all these new data modalities that we're going to be getting.
Mm-hmm. Yeah, that's a good point. So I'm going to put that down as a action item as well. That's something that's pretty simple. It's just a matter of sharing for the consortiums that already have something there, just simply sharing it. That sort of, maybe that kind of counts as the inventorying step, but so I will follow up with you guys, I guess, about that, and then I'll talk with the other person who works with me, who's also part of HCA, to see if he can get me to, maybe we can at least start with this set of consortiums and see if we can along the way get others to share theirs as well.